In this lecture, I'll review the key characteristics of yeast that are relevant to brewing beer. I'll actually begin with an overview of the range of organisms usually involved, and that'll take us beyond Saccharomyces into molds and bacteria, but we'll return to fundamental aspects of yeast metabolism. In particular, I'll review the regulation of yeast metabolism and how that regulation is relevant to ethanol production and to brewing generally. And I'll hint at how metabolism is relevant to flavor, but we'll cover those molecules and their origins in another lecture. I'll then focus on flocculation, the clumping and precipitation of yeast cells, as a variable that brewers focus on, with an emphasis on molecular mechanisms of flocculation. I'll point out how, though we tend to make generalizations about the flocculation behavior of yeast, it's actually a very complex problem that depends not just on the strain of yeast, but on brewing conditions as well as the reproductive age of the yeasts. That fact that brewhouse conditions can affect yeast brings the lecture to a close with a really important brewing history story, how the traits of yeasts used in quintessentially European beers are actually products of accidental brewhouse selection, uniting wild New World yeasts with brewers' yeast strains. My lecture style involves detailed digressions into biochemistry and molecular biology. I hope you find the way they enliven what would otherwise be a standard review of the key traits of brewing yeast: attenuation and ethanol tolerance, flocculation, temperature dependence, and sensory profile. Even though we refer generically to brewer's yeast, we're usually referring to one of two distinct species, Saccharomyces cerevisiae and Saccharomyces pastorianus. We refer to these as top fermenting and bottom fermenting respectively, as the top fermenters tend to rise to the top of the vessel, forming a krausen or crust of cells. After attenuation, that is, after the consumption of glucose, both types of yeast flocculate, that is, aggregate, and fall to the bottom, though this is a trait that varies widely among yeast strains and even within a particular strain. Another interesting feature that we'll return to at the end of the lecture is that the brewhouse places evolutionary constraints on yeast that have downstream effects on brewing. Top fermenting yeast, for example, were often harvested by top cropping, or collecting the yeast from the krausen. In this way, the brewer automatically selects for yeast that's good at forming that crust. Modern brewers use conical fermenters since, after attenuation, the yeast falls to the bottom of the vessel. A conical bottom means the yeast plug, or tube, can be withdrawn easily for repitching. This obviously alters the selection landscape of the brewer, since only flocculated yeast is collected. Other yeasts are of interest too. Some yeasts are acid formers, like the spoilage organism Brettanomyces. Of course, one person's off flavor is another person's style parameter. The otherwise gross, mousy flavors generated by this species are important in Belgium and Lambic beers, where this yeast appears as a secondary fermenting organism, secondary to Saccharomyces cerevisiae. Other relevant bugs are molds. In fermented rice products, Aspergillus provides the amylases required for starch mobilization, though Saccharomyces cerevisiae is still critical for ethanol production. Yet other fermentation-relevant organisms are bacteria like Lactobacillus, which produces the lactic acid that turns milk and yogurt sour. And there's also Pediococcus, which among other things generates diacetyl or diacetyl, a strong off flavor. Often this large, diverse group of organisms are referred to collectively as LAB or lab, lactic acid bacteria. To place all these microbes in context, let's look at a simple phylogenetic tree of life, showing the relationship of our key organisms. Pediococcus, Leuconostoc, and Lab generally are all gram-positive. They possess a peptidoglycan layer on their exteriors that stains purple in crystal violet, while their gram-negative bacteria, which lack that structure, do not. Just for comparison, E. coli lacks the peptidoglycan layer, is a gram-negative bacterium in the phylum Proteobacteria. E. coli would definitely be considered a spoilage organism, but does show up in some wild fermented beers, as we'll see. The star of the show, yeast, is a fungus and falls within the eukaryotes, organisms with cell nuclei, and are more closely related to animals than plants. Taking a closer look at fungi, we see things get really complex. While the evolutionary relationships are still clear as we trace the tree out from a common ancestor to the branches of individual groups of organisms, recall that these trees can be constructed from a variety of data: physical traits, amino acid sequences of proteins, but perhaps most definitively by the ribosomal RNA sequences, since those are among the oldest and slowest changing nucleic acid sequences and are present in all life forms. For reference, mushrooms reside in the phylum Basidomycota and the subphylum Agaricomycotina. Molds, but also Brett and Saccharomyces, are also fungi residing in the phylum Ascomycota and subphylum Saccharomycotina, along with the zombie fungus Cordyceps, which is not, so far as I know, used for brewing beer, though one might argue over whether we control the yeast or whether we, like the ant in this picture, are actually controlled by the alcohol-producing fungus. Focusing on the key traits of yeasts, strains may all be described by their individual tendencies to tolerate higher or lower concentrations of ethanol. And that's related to their ability to attenuate or convert more glucose to ethanol, to flocculate, which is a characteristic that shows up after attenuation, their cryotolerance, again, Saccharomyces pastorianus is content fermenting at lower temperatures, and their production of different flavor compounds. I keep coming back to this slide, but it's important to remind ourselves. Complex carbohydrates in this case means a disaccharide. Other, more complex carbs are unavailable for fermentation. The action for brewing beer is really the reduction of acetaldehyde by NADH to NAD+. The NAD+ allows glycolysis to continue while oxidative phosphorylation is shut down under anaerobic conditions. In the case of lactic acid bacteria, pyruvate is reduced by NADH to lactic acid, producing NAD+ for the same reason. Interestingly, in acetic fermentation, when making vinegar, say, acetobacter runs the other way. In the presence of oxygen, ethanol becomes a substrate for reduction of NADH to support oxidative phosphorylation in the absence of glucose. Now let's anchor ourselves in biochemistry. Take a look at what's happening in enzymes like alcohol dehydrogenase. NADH is the reduced form, the reducing agent. NAD+ is the oxidized form, the oxidizing agent. In alcoholic fermentation, NADH is oxidized to NAD+, with its proton and electron going to reduce acetaldehyde to ethanol. As a side note, NAD-based dehydrogenase enzymes like alcohol dehydrogenase are very common. There are more than 500 spread across many pathways and functions, and still more that are not dehydrogenases, but oxidoreductases that require cofactors, similar to NADH. So much for ethanol production. Flavor is also crucial to brewing, and many other molecules besides sugars play a role. So while we're discussing metabolism, let's notice that the flavors we'll encounter have their origins in energy metabolism. Fuels come into the cell not just as carbohydrates, but as fatty acids and amino acids, which pass through the usual suspects of metabolic pathways. This figure endeavors to place these pathways in the context of metabolic flavor endpoints: esters from fatty acids, fusel alcohols and aldehydes from amino acids, and the keto acid pool. We'll come back to each of these in a future lecture. The keto acid pool is really the set of components in the tricarboxylic acid cycle, or Krebs cycle, or citric acid cycle. Remember, we usually think of the TCA cycle as a generator of NADH for respiration, but those intermediate acids form building blocks for a range of other endpoints. The foundation for all those other pathway choices is the way yeast responds to the presence or absence of glucose. Catabolite repression describes a system by which the molecules of metabolism, glucose, affect a set of enzymes made by the yeast to repress or activate the use of those other fuel sources. This diagram is an overview of some of the key players, the regulatory proteins of that system. It's a feedback system that allows the cell to respond differently to different nutrient conditions by changing the presence or activity of proteins involved in metabolism. One kind of regulatory protein is a kinase that adds a phosphate group to change the behavior, activating or inactivating some target protein. In In the glucose sensor pathway lies SNF1, a kinase that alters the behavior of MIG1. SNF1 is itself phosphorylated by another kinase, SAC1, a glucose sensor. Under low glucose conditions, SNF1 is activated, that is, it's phosphorylated by SAC1, and it goes on to shut down MIG1. That means MIG1 is phosphorylated by SNF1, which leads to derepression of gluconeogenesis, that is to say, the activation of gluconeogenic pathways. Low glucose, therefore, means yeast can make glucose. Notice that in one case, phosphorylation activates the kinase, SNF1, and in the other, it inactivates the kinase, MIG1. Also, activated, that is, low glucose, SNF1 derepresses alternative carbon sources and stress response systems in addition to gluconeogenesis, so that the cell can turn to those other carbon sources, say fatty acids, as a fuel source. Conversely, under high glucose conditions, SNF1 is inactivated, dephosphorylated, allowing MIG1 to repress glucose synthesis. Why do we care? Well, yeast's metabolic state is reshaped genetically according to conditions. Nutrient availability and metabolic activity or condition can affect the kinds of molecules that get formed, and these molecules will impact flavor, as we'll see in a subsequent lecture. Likewise, a cell remodeled for fatty acid consumption because of glucose depletion, when repitched into sugary wort, can result in a stuck or stalled fermentation. So while the metabolic state is plastic, it can persist for long enough to affect future fermentation outcomes. The Crabtree effect, first described in 1928 by British biochemist Herbert Crabtree, describes how yeast switches from ethanol production to ethanol consumption under aerobic conditions, and lowering biomass production as well. The preference for energy production over biomass accumulation is a key trait in yeasts used in industrial biofuels as well as alcoholic beverage production. The idea is that switching to ethanol lessens the organism's dependence on oxidative phosphorylation so that they might outcompete other microbes trying to access the same glucose supply. It's worth pointing out here that yeast is a facultative anaerobe. It can grow in the absence or presence of oxygen. The Crabtree effect is observed under aerobic conditions. Under anaerobic conditions, ethanol production is not turned off, there's no dioxic shift, and there's no consumption phase, though spoilage bacteria could oxidize the ethanol to drive their own energy production and make acetic acid. For comparison, we see on the right the Crabtree negative Cleveromyces, which does not produce ethanol, but does have alcohol dehydrogenase enzymes for consumption of ethanol as a fuel source. There are 7 alcohol dehydrogenases in yeast, generated either through single gene or whole genome duplications. This shows another phylogeny with the timeline of evolution of alcohol dehydrogenase in yeasts over the last 200 million years. ADH1, it's called 1 even though it's the last to evolve, is primarily responsible for ethanol production. It's active against all alcohols between 2 and 10 carbons, though the Km decreases with the chain length. This trait emerged after a whole genome duplication and a subsequent gene duplication, which allowed for the selection of a new trait of high acetaldehyde to ethanol conversion. What's the dinosaur story? Well, really, it's the emergence of fruit-bearing plants that happens around the same time as the mass extinction that swept away all the dinosaurs. It's likely that yeast evolved this trait as a means of, on the one hand, taking advantage of the new sugar supply, and on the other hand, producing a poison that would allow it to outcompete other microbes. ADH2, on the other hand, is a product of a much earlier duplication. The enzyme is only produced under aerobic conditions and is used for the consumption of ethanol as a fuel source. ADH1 evolution happened after Cleveromyces diverged, so Cleveromyces only has ADH2 activity and doesn't make ethanol, though again, it can use it as a fuel source under some circumstances. Ethanol tolerance versus high production ability is a very complex trait associated with stress tolerance. Tolerant strains adjust membrane composition and upregulate heat shock proteins that can help it adjust to the more challenging high ethanol concentration environment.